Okay, as you know that plasma cell will secrete antibody, but how can antibody work against the pathogen? So we first need to know the structure of antibody. Antibody, there's one form of antibody called immunoglobulin. Immunoglobulin is one type of antibody. They are Y-shaped. And then they are made of a heavy chain and light chain. On the antibody, you have a constant region and variable region. The variable region, they are the one that binds to the antigen. The constant region, they are the one that doesn't change. That's why we call it constant. Variable region binds to antigen because we have different types of antigen. Many different. That's why the variable region can be changed. One way you can memorize that variable region is the V. What does it mean? It's the V. You see that variable region is the V. So you can memorize that variable region is where this the V shape here. It is where the antigen binds to it. And the constant region is basically here. And you have heavy chain and light chain. This is the real this is I should say this is electron micrograph for the antibody. Okay, there are 5 classes of antibodies. Yes, they all have their different function and distribution. In Bio 3, I will not ask you the function and distribution, but if you take my physiology, I will ask you this function and distribution. One thing I want you to know is that we have 5 classes of antibody. You need to know the label. A, D, E, G, M. So these are the 5 classes you need to know. A, D, E, G, M. That's all. Okay, how does T-cell works? So T-cell, they will leave thymus after they mature in thymus, and then they will get the T-cell receptor. So how does it work? T-cell cannot recognize the antigen directly. It needs someone to help him. So what happens is that if you have a pathogen here, T-cell cannot go directly to use the T-cell receptor binding to the antigen or pathogen. What happens is that your macrophage, they have to kill the pathogen first. After the macrophage use the phagocytosis to kill the pathogen, then the phagocyte will express the antigen of the pathogen on the cell surface of the macrophage. And how does it express? It uses the MHC protein. It's called MHC2, not MHC1. The macrophage will hold the antigen from the pathogen. The MHC2 protein is like it will be expressed on the plasma membrane on the macrophage, and this MHC2 protein will be holding the antigen of the destroyed pathogen. Then in this case, the T cell can bind to this MHC2 protein complex and to recognize the antigen. And what I'm talking about is the helper T cell here. And then you have another type, which is called cytotoxic T cell. Helper T cell is the one that usually is the one there to recognize the antigen, and then you send out a cytokine like interleukin 2 or interleukin 1 to other cell to tell the cell to other immune cells so that they will respond to it. Cytotoxic T cell, as the name suggested, you will secrete cytotoxin to kill cancer cell or infected body cell. Okay. So basically, this is what I just told you. So you have T cells. What happens is that you have a dendritic cell, so it's another form of cell that can do phagocytosis. It phagocytizes a pathogen, and then after it breaks down the pathogen, then it will express the antigen. The green dot is the antigen on top of MHC, and it should be MHC2, not MHC1. MHC2 protein, and then, because see here it is MHC2 protein. But here it says that MHC1, so it should be MHC2 protein. And then the T cell will bind to the antigen MHC2 protein here. Now after this, then the T cell will be activated and it will clone itself, so that all these T cells will have the information of the antigen. It will secrete cytokine to tell other cell to it will secrete cytokine to other cell that to tell other cell that okay you have to tell other white blood cell to about the information of the pathogen. So the other white blood cell can respond to it. For example, the B cell, after they receive the cytokine, they will start to clone themselves, and some of them become plasma cells to secrete the antibody. T cell, some of T cell, they will also go through apoptosis when the infection is over. Some of the T cell will become memory T cell, so that when the next infection occurs, they can immediately start to secrete a cytokine. So this cytotoxic T cell can kill cancer cell or virus-infected body cell. What they do is a two-step process. What they do is that they will secrete perforin. Perforin acts very similar to MAC. Remember MAC, membrane attack complex? So perforin will form a pore on the cancer cell or virus-infected cell membrane, and then after it forms a pore, instead of letting the cytoplasm to leak out, the cytotoxic T cell will secrete granzyme. Granzyme is a cytotoxin. So basically, it make a hole on the cancer cell, let's say cancer cell, and then it will dump the cytotoxin, granzyme, into the hole, so that it will literally poison that cancer cell, because the granzyme causes apoptosis. And then some of the cytotoxic T cell will also become the memory T cell. So that next time when the same immune response occurs, then the cytotoxic T cell can immediately jump into action. So this is what I meant. This is our cytotoxic T cell. This is the target cell. Can be cancer cell, can be infected body cell. So you will secrete perforin to make a hole, and then you will secrete a granzyme to go through the hole made by the perforin, so that the granzyme can go into the target cell and kill the target cell. It looks like this: cytotoxic T cell and target cell